How often do you hear about microorganisms in the news? About viruses, bacteria, fungi? Some microbes, like the SARS-CoV-2 virus that causes COVID-19, are notorious in the news and social media for being agents of infectious diseases. These often lead to conditions that threaten our immunity. However, not all microbes are harmful. Out of the billions of species of microorganisms, only some are identified as virulent or being disease-causing, whereas most microbes, like the probiotic bacteria that we eat in yogurt, are actually quite beneficial for our body. Synthetic biology merges the principles of engineering with the concepts of biology and the life sciences. This way, we can harness the potential of microorganisms as novel biological systems that can accomplish a myriad of functions.
So why should we use synthetic biology for clinical or medical applications? Firstly, synthetic biology-based systems can be advantageous as diagnostic tools, as they can potentially overcome the limitations of traditional methods, such as antibody-based platforms, which can tend to be more costly as well as time-intensive and labor-intensive to produce. Additionally, through Synbio, we can make diagnostic tools that are easier to interpret through the use of biomarkers, which are basically substances in organisms that can be measured to indicate disease. In recent years, MIT and UCSD published a paper that explores how simple Synbio techniques may be applied to genetically engineer microbes for the detection of cancer. Specifically, the non-virulent E. coli strain, Nissle 1917, was genetically modified to include a lac Z operon in specific gene circuits, which are basically assemblies of biobricks and biological parts which encode RNA and subsequently protein in order to enable functional outputs. The modified E. coli strain was able to produce a luminescent signal as the biomarker in urine. This modified Nissle 1917 was orally administered to mice and was found to only colonize the liver. The lac Z enzyme, which is encoded by the genes in the inserted operon, cleaves LuGao, which is essentially luciferin binded on galactose, to release free-floating luciferin, which is then used as a biomarker and detected in urine. The presence of this biomarker in the urine indicates the colonization of the Nissle 1917 in the liver, and thus indicates the presence of a tumor in the liver, either due to liver cancer or a tumor from another cancer that has metastasized. This is only one of the thousands of biomedical applications that genetic engineering has been used for. Many of these products and applications being developed by fellow iGEM teams in the diagnostic and therapeutic project tracks. In a post-pandemic world, we hoped that investment in biomedical technology will increase the number and success of these breakthroughs, from the development of readily deployable diagnostics to the building of microbe-based vectors used in therapeutics.